So I'm Surag. Nice to meet you, all of you. Thanks for coming. As introduced, I'm a machine learning scientist in Gokchan's group at Genentech, and I'll be presenting our work on that we call Nona. It's a model for unifying functional genomics. That's how we see it through multimodal masking. And I'll just set up what that means. But before that, I'd like to thank all my amazing colleagues at Genentech who helped me through different aspects of this work. So as Gokchan alluded to in his awesome primer, you can kind of split the main categories of models for regulatory DNA into broadly these three subsets. The sequence to function category, which has seen a lot of interest in the last few years, where you make predictions from sequence alone. And as Gokchan mentioned, there are models that take in starting from about 500 to 1,000 bases to models like Informer and Borzoi that take in DNA sequence windows as long as half a megabase in some cases. There's also variations of these frameworks where instead of predicting from sequence alone, you might predict from sequence plus one or other functional tracks. And one example of this could be where you're trying to predict gene expression in a specific cellular context from the DNA sequence and the attack-seq, or some notion of chromatin accessibility in the same cell type. And then more recently, what's really exploded is language modeling, both autoregressive and masked language models like DNABird, Caduceus, Evo, Evo2, a nucleotide transformer. There's all kinds of different ways in which people tokenize, people design the masking objective, autoregressive versus masking based. Also, what species you're training on can be completely different species, can be different individuals within the same species. A lot of different modeling challenges that are involved in training these kinds of models. So what we started thinking about, and what Gokchan mentioned, was that all of these are being developed in silos almost, especially like language modeling and sequence to function models. And if there's any way to start unifying these models, if you were to train today a language model, you'll have to go to a completely separate part of the internet or of GitHub, I guess, and set up a bunch of tools and then get started with language modeling. You're going to do sequence to function. You'll do a completely different set of data processing and training for these. So as we start thinking about the process of unifying these almost disparate modeling approaches, we realized that in the process of unification, we can actually start doing things that are actually none of these. We can start going beyond these highly siloed classes of models, and we can do something that's more interesting and spans and is almost some kind of hybrid or hybrid approach that you wouldn't classify into any one of these categories. So maybe to motivate that a bit further, I want to show you an example of a text vision multimodal masked model. This is one of the places where we draw our inspiration from. And the way these models work is you have pairs of images and captions, and you can think of arbitrarily masking patches in your image or tokens in your text and then filling those in. And the model is able to look across modalities and look at correlation between the text and the images and fill those in going across modalities. And what's nice about this kind of framework is it captures the more common separate models that you see, things like captioning an image, right? This is an entire class of models on its own, but is encompassed in this broader framework of multimodal masked modeling, where you could, for example, train a model by simply masking all the text tokens and predicting them conditioned on the image. Or you can go the other way around, where you try to generate an image given tokens in the text, right? But you can also go beyond this. You can do some interesting things where you're editing the text, you're masking specific patches of your image that help you edit the image itself. So it's not exactly captioning or image generation. It's some kind of an in-between task, but also interesting in its own right. And I think if you've tried giblifying your images, you kind of know what I'm talking about from the last week. So this is where we draw our inspiration from. And coming back to functional genomics, you can really think of two broad modalities. The first is DNA sequence, and the second modality is all our functional tracks, although you may also wish to call RNA-seq a completely different modality from attack-seq, which it probably is. But for now, you can think of it broadly as DNA sequence and all your functional tracks. And what's nice, and it's a little different from the text and image case is that these share a common coordinate system, right? So when you do a tag-seq or RNA-seq, you get reads, you align these reads to the genome, and then at every position in the genome, every base, you get some read count for your assay, right? So it's really nice, and you can represent it in this matrix form where you have sequence, typically one hot encoded, for channels for A, C, G, and T, and then the tracks you're considering as a one-dimensional vector with the same length as the length of the sequence and nicely aligned for you. So the crux of Nona really is a multimodal masking-based model where we have our matrix of different functional tracks and DNA sequence. And in theory, we can arbitrarily mask individual pixels both across the tracks or the tasks and the DNA, and then kind of jointly fill in the missing pixels based on what else you see. Right? So this is seems to be like a crazy flexible approach, which it is. And I'll try to now make more sense of it in the next few slides. But I hope you can start to see how this is already unifying different approaches in one larger class of models. Great. So you can now start thinking of how you can encode the first, three different classes of models I've shown you within this single framework. And then I'll also show you how we can come up with entirely new classes of models or types of models that don't fit neatly into any of the first three buckets I've shown you. Right? Operationally, the way Nona is working is we have our data matrix, and we mask out positions in our data matrix with zeros. So if there was some non-zero value, it becomes 0. If there was a base there, there will be a base there, or unless, I guess, there's issues with the reference genome, you convert that into zeros with an n. And then in addition to that, you also append a mask matrix. It's just a binary matrix of ones and zeros, where it's 1 where the specific pixel was not masked in the input. The reason for doing this is so that the model can differentiate between zeros in the data matrix caused from masking versus zeros that are actually observed at this specific position. So sequence to function modeling now just becomes an instantiation of a Nona model where you mask all your tracks and you keep your sequence unmasked, right? So a very simple case where during training, what you can do is have a bunch of tracks, you mask all of them out, and you only predict those from sequence alone. If you want to add more tracks to your sequence, you can simply do this really in a dynamic fashion, right? You can dynamically unmask different tracks when you're training or during inference, and then predict the rest. So you can have perhaps sequence plus a tag-seq in skin cells, and then try to predict the gene expression in skin cells or all other cell types as well. Language modeling, in the case of language modeling here, what you could do is either remove all your tracks, or if you have some tracks, you can completely mask them and then ignore them in terms of your loss function. And what you then do is just mask at specific bases here. And this kind of recapitulates your masking, your masking-based language modeling. So pretty straightforward, but now that we've set this up, we can actually do a lot more than just these three things, right? I'm going to show you some examples of things that we've thought about that have been specifically motivated by downstream applications in functional genomics. So one thing we've been thinking about is what we call context-aware prediction, in which the input of the model is not just the sequence, but also the functional measurements adjacent to some region of interest. And I'm going to talk about this specific mode in more detail in the next section. But just to set this up, there can be use cases where you might want to factor in information beyond just the sequence itself. We have all our tag-seq and RNA-seq measurements across cellular contexts. Other ways to make better use of them when you're trying to make predictions in a region of interest. Another interesting mask we thought about is what we call positional masking, where we mask out these functional tracks at specific at bases at random. And what this kind of a mask can help us potentially is filling in what's these functional tracks at these specific positions from the sequence but also the functional measurements at other bases. And you can think of ways where this might be useful for denoising tracks, especially those that have either high noise or low read depth. And we can also extend traditional sort of vanilla masked language modeling into what we call functional language modeling. Here, instead of completely ignoring all the functional tracks and the wealth of data we have, we can actually factor those into our masked language modeling approach. So instead of masking out all our tracks, we also provide the model access to our tracks, only mask out specific bases, and fill those in not just from the sequence, but also from the tracks that we observed. But hopefully, I'm starting to convince you that this is a fairly flexible, versatile approach, almost a Swiss Army knife of sorts of functional genomics. If you have an interesting application in mind, you can come up with a nice masking strategy and train a model that hopefully tackles the use case you have in mind. I'm going to pause here, but what I'll be doing in the next few minutes, I guess the next half an hour or so, is walk through 3 completely different use cases of Nona, in which we'll be training 3 very different kinds of models. So it's almost like 3 different flash talks unrelated from each other. So hopefully you can get a sense for the versatility and breadth of the approaches we have. But good time to ask questions, if any.
Did you try training all 3 tasks together? And can
Yeah.
you see generalization across different tasks when you do that?
It's a great question. So we have, in today's talk, I'll be showing you cases where we just train on one kind of mask at one time. We have experimented with training multiple kinds of masks in the same model, and that does work to some extent. So far, what we've seen is that if you want optimal performance on any one specific kind of masking, it's better to either fine-tune on that or train exclusively on that. I don't think we have seen some instances where we can generalize across masks. So if you have a mask that you haven't seen during training that is somewhat related, there is some sense of, but we haven't really gotten into that into much detail.
So in the kind of multimodal, like vision, text stuff, people, many models like tokenize the modalities separately. I guess you'll talk more about that kind of thing. And do you see that as the tokenization, et cetera, being constant throughout these different sections? Or do you think it like depends on those?
Yeah. So I think, as I mentioned, we're sharing a common coordinate system, which almost makes it easy to represent what's going on locally within a genomic window. And I think they're just keeping in spirit with the kinds of models we've seen in the space, where we're just either doing sequence to function prediction without any kind of tokenization, predicting from raw DNA sequence itself. It's an interesting, we've thought about it, but yeah, I haven't explored that in mind.
Might be jumping the gun a bit, but we heard that in sequence to function models, they're somewhat limited by the counterfactuals being on the sequence, the perturbations. Is another motivation for this model also being able to perturb the tracks and seeing changes?
Yeah, exactly. Yeah.
I think it's a bit related to Salil's question. I mean, there's not an implicit tokenization in Informer or Borzoi, but the convolutions do that and reduce the size to 128 base pairs, right? Where there's the scale, the functional scale where, I don't know, ChIP-seq might vary. I mean, doesn't that constrain a lot the size of your sequences and the size of your inputs? So I guess it would kind of make sense.
Yeah.
Maybe.
So the models I'll be showing you today, some of, especially in the first subsection, in the first section, are really long, about 200 KB, and we're able to model these at base resolution. So within our unit architecture, we can go all the way back to single base resolution. And you're right that there are some constraints. We probably can't model a few thousand or tens of thousands of tracks, but in the range of a few hundred, it's quite doable. Great, so I'm gonna move ahead into the first section. Thanks for the questions. Into what we call a context-aware Nona model, right? So to set this up, sequence to function models predict these assays like DNase-seq, ATAC-seq, RNA-seq from sequence alone. What if we provided the models more information? Could there be cases where this might be useful? Especially if there's some event of interest that's somewhat localized, some kind of integration or a mutation, for example. So maybe one way to think about it is, let's say you have a reported gene of interest and you know you're going to integrate it at some specific locus in the genome, but you want to predict how much once upon integration, what the expression level of this gene is going to be, right? And so this would be a function of the promoter sequence of the gene and also the genomic environment or the sequence and the accessibility and other factors in the chromatin state of the locus into which it's integrated, right? Other enhancers may interact with the promoter once it's inserted into this specific genomic coordinate. The sequence to function way to make this prediction would be to take the sequence of the site of integration, insert the sequence of the reporter gene, and then run it through a model like Borzoi or Informa and look at the predicted RNA-seq or cage values in the cell type of interest, right? But the hypothesis we have here is, can we do better if we add additional information? So we already know something about this specific locus beyond the sequence itself from our measurements. So we perhaps know that there's an attack-seq peak 20 kb upstream of this and another one 80 kb downstream. Is there any way to factor this in this specific, make these kinds of predictions, right? So to test this, we trained a context-aware model for K562, the K562 cell line. In this model, we have 9 tracks, 7 DNase-seq tracks, and 2 cage tracks, which is a subset of the larger set of tracks that Informa works with. And we train our model in 2 stages. In the first, this is all done within the Nona framework, so you can just switch the masks up at different stages of your training, really. So in the first stage, we're doing a sequence to function training with a 196 kb input window and 9 tracks. And these 9 tracks. In the second stage, we fine-tune this model with our context-aware masking mode, in which we're masking only the central 4 kb. And so the model has access in these predictions to the sequence and 192 kb of measurements around in regions adjacent to the central masked area. And it's trained on the same genomic regions, so both of these models are trained. The only thing that changes when you go from part 1 to part 2 is the masking, right? Nothing else really changes. The loss and everything else stays more or less the same. And so what we can see here is the prediction on the test set for these 9 tracks. When you're predicting with just the sequence-only mode of Nona or the context-aware mode of Nona. And you can see across the board, pretty decent increase in performance for all the DNase-seq prediction tasks as well as the gene expression cage tracks, which suggests that, as you'd expect, adding the context information does give you a bump over the sequence only.
I just curious about what happens if you don't include the sequence. So if just context only prediction, are these better, or what are the numbers here look like?
Yeah, don't try that explicitly. I would imagine it's somewhat lower because you would need to know, for example, if there's a TSS of a gene in the central masked region, right? If you don't have access to the sequence, you wouldn't even know if there's a TSS of a gene, so you may not be able to predict gene expression in the first case. So my expectation is it'll be fairly low. Great. So when we look at the differences between the predictions of these 2 models for one specific track, in this case, DNase-seq and K562, you can see that the predictions are more or less tightly correlated, except for this set of about 2% of regions where the predictions of the context-aware model differ significantly from the sequence-only model. In fact, in this case, are much lower than the predictions of the sequence-only mode. And this one specific locus where we see this phenomena. At this specific locus, the sequence-only model predicts kind of across the board high regulatory activity. So it thinks there's a strong DNase-seq peak, chromatin region of accessible chromatin here, another one here, and another one here. But if you look at the actual data, and this is a test set example, so if you look at the actual measured data, you see there's no regulatory activity here, very little regulatory activity in the other positions, and also in the central 4 kb. So if you kind of zoom into the central 4 kb, you can see that the model really thinks there's a lot going on here in terms of chromatin accessibility, but there just isn't as much going on. And so what the context-aware model will do in this case is have access to the same sequence as the sequence-only model, but also look at the fact that there's overall reduced accessibility in the adjacent flanking areas. And so when we use the context-aware model, it seems to reduce its prediction compared to the sequence-only prediction. Although not completely ablating it entirely, it does seem to move towards the right direction. And for those of you fairly familiar with chromatin states and the like, what we see is that this specific locus seems to be enriched for heterochromatin marks. These are just chromHMM state tracks for a bunch of K562 samples, and see a lot of enrichment for heterochromatin and ZNF repeats. But overall, we seem to, when we do this analysis more globally, we do see an enrichment of heterochromatin or other repressive marks in region where the context-aware model's predictions differ from the sequence-only model. So we try to do a different kind of evaluation, one that reflects how we'd like to use this model for a downstream application. And the kinds of data sets that are a good fit is something called ChIP-seq. I think it's called thousands of reporters in parallel assay. Yeah, that's why it's called ChIP-seq. And the way ChIP-seq works is you have some promoter and reporter gene construct that you insert randomly basically at different loci in different cells, somewhat at random. And then you measure the expression of the promoter and reporter gene once it's been integrated into those contexts. And so the differences in expression would largely be driven by the chromatin state or the genomic environment within which this gene was integrated into, right? And so what we end up at the end of this experiment is the coordinate of integration and the gene expression measurement for this reporter gene, right? And people typically integrate the same promoter in a few hundred to a few thousand different positions, make these measurements, and then also repeat this for a bunch of different promoters. So housekeeping versus a more cell type specific promoter. And the way we would run these through our models is simply, the reporter's sequence is about 2 kb in length. So we can integrate that, we can insert that into the sequence of interest at the site of integration in the case of a sequence-only model. In the context-aware case, we will append the measured functional tracks outside the 4 kb in the center, which will contain 2 kb from the reporter sequence itself. So here the model is seeing the sequence post-integration, and then also seeing prior to integration what the chromatin landscape looked like. And we're kind of assuming that that doesn't change much after integration. And so what you can then do is look at the prediction of the models with and without the context information. So for this one specific promoter that was integrated in about 600 sites in the genome, you get a bunch of gene expression measurements, and then you get predictions from both our models, and you can look at the correlation between the measurements and the prediction in both of these cases, and we see reasonable increase in accuracy going from the sequence-only mode to the context-aware mode. And you can repeat this for a bunch of promoters, and we see a similar pattern on average. There are some promoters that we don't seem to do better on, but there are others where we see reasonable gains on. And then you can also repeat this with another more recent data set, and you more or less see the same trends where adding information about the context seems to help you predict the expression levels of integrated reporters better. So that was the first flash talk, I guess. And I've hopefully shown you that a context-aware Nona model can integrate information beyond the sequence and specifically integrate information about functional measurements around the central region of interest. Great. So we're going to switch now to functional language modeling. And as I mentioned before, what we're doing in this mode is extending masked language modeling by conditioning on additional functional tracks. In this case, let's start off with a simple one-track example where you have measurement from one track, let's say a tag-seq or DNase-seq in one specific cell type. You mask some fraction of bases, then you fill those in. In all of these cases, you're presenting the entire track to the model, but you're only masking some bases in the sequence, doing this at single base resolution. And so we started training some of these models to see what they're learning, and then what we could do with these kinds of masked language models that are aware of, that are functionally informed. And so we started off with a 512 base long model, fairly short. And the track of interest in this case was DNase-seq in GM12878, which is a blood-derived cell line. And what we do during training, which is a little distinct from how models like BERT are trained, is we used a uniform masking rate. So uniform between 0 and 1. And so in some examples, you might mask as high as 90% of your bases. And then some you might mask as low as 5% of your bases. And I'll show you why this is useful when we talk about how we can use these models to generate regulatory elements. So the first question we have is what are these functional language models learning? Are they even different from what masked language models are learning? And if we can make any sense of them. So the simplest thing we thought of was to just mask one base at a time and then look at the model's predictions for that base. So conditioned on some profile of DNase-seq, in this case, GM1287 and DNase-seq. Let's say we know this region is an accessible region in GM12878. And given everything except one base in this at this locus, what do the model's predictions look like? And we'd like to differentiate between two major cases where the model is kind of unsure, or it's just giving you the baseline distribution of A, C's, G's, and T's, versus where the model is quite confident of the base at this position. So whether it's a flat distribution versus somewhat peaky. And we can quantify this by just looking at 2 minus the entropy at this site, which would be higher for cases where distribution is more peaky and lower where it's flatter. And the entropy here is just the standard negative sum of P and P log P. And so the way you would run this interpretation analysis then would be for the same locus, so the same track and the same sequence, you mask one base at a time. So you go across the sequence, masking one base at a time. You compute this 2 minus entropy value, and then you just concatenate them all together to get some sense of what positions in this input sequence is the model paying more attention to, or has stronger predictions, is making more confident predictions about. And so I'm showing you one specific 512 long sequence, which you can't probably read, which is totally fine at this point. And then the GM12878 signal. So you can see there's a peak. So this specific sequence comes from an accessible chromatin region in GM12878. And now what you can do is scale each base in this very small sequence or very narrow sequence by the 2 minus entropy values that I showed you before. And when you do that, it kind of it clears up, right? So you see higher scores kind of localized, almost DNA words in some sense, shooting up. And we're just centering the 2 minus entropy scores, subtracting the mean to make it a little more clean. Now I'm going to zoom in now so you can finally read the sequences, especially those of you that can recognize transcription factor motifs by eye. It's a good time for you. But what we're seeing is that the model is picking up on specific DNA words. And I'm going to show you, actually, these are well-known features of DNA, and these are quite well annotated. I'll just tell you what these are. So the first one is actually a simple repeat. It's a microsatellite. And you can see the sequence is a GCC repeat, about 7 or 8 times. It's been repeated. And then it makes sense, right? These kinds of simple repeats are scattered through the genome. If I just mask one base, if I just mask this G here, it's going to be pretty easy for me to predict that, yeah, there should be a G here because I'm seeing the GCC pattern repeat on both sides, right? So that very simply explains why the model is able to be more confident in its predictions at this microsatellite or this microsatellite sequence or motif, if you will. But this is just like some kind of idiosyncrasy of the genome. It's not really functionally relevant. It's not that it's a transcription factor binding site in this case that's causing the region to be accessible in GM12878. But what's even more interesting is these other DNA words or motifs actually correspond to known transcription factor binding sites. So this GCCC and GCC is an SP or a KLF motif, and these others are NFY motifs. So these are quite well annotated.
So have you compared these motifs that are picked up here versus the motifs that would be picked up with the gradient times include method, and how do they compare and how consistent?
I don't have the slides here, but yeah, the short answer is you see these SP and NFY motifs, but not the microsatellite.
And do you have any intuition as to why?
So yeah, I mean, the SP-NFI motifs are known to cause accessibility, right? So they are associated with increased accessibility. Having this motif means that a transcription factor is binding there and is bringing other transcription factors and keeping the region open and accessible. There's also a promoter sequence actually, so you can see. So if you look at a sequence to function model and make predictions with it, you're going to see mostly these motifs, but not the microsatellite.
I also have a related question, which is, do you see what are some of the motifs that like you feel like you can't actually like learn and uncover? Because obviously there are things that are easy to find and there are things that maybe are a little bit harder.
Yeah, I mean, I don't think they're specific to these language models, but there are certain zinc fingers, for example, that don't bind a lot. They maybe bind tens or a few hundreds of sites. I think those are some kinds of sequence features that you wouldn't be able to learn just because it's a very rare event. But any transcription factor that's fairly pervasive, binds, promoters, or enhancers, you should, and we have seen in many contexts, you are able to pick with these models.
Given that, like the DNA language models are like zero-shot predictors of,
yeah.
Have you tried doing this entropy, but with just the...
Next slide, yeah. Thanks. So yeah, we looked at Caduceus, which is a masked DNA language model, completely unconditional. And what's interesting is it does pick up the microsatellite, but not the transcription factor binding sites, which somewhat makes sense. These are, well, you would, so we do know that these DNA language models pick up features like start codons and sometimes even a CTCF motif, but don't really pick up much beyond that. And yeah, one reason for that is they aren't really informed by any functional measurements. And as Gokchan mentioned, the transcription factors relevant in different cell types are different, and they aren't necessarily under strong constraints as much as, say, a start codon is or genic features are. And so I think that might explain why a language model like Caduceus is not able to pick this up. So once we've established that our model is learning meaningful features of DNA, not just the idiosyncrasies like microsatellites and other repeats, but also motifs, we wondered if we can repurpose this masked language model for designing regulatory elements. And as Gokchan mentioned, synthetic regulatory element design has kind of taken off in the last few years. Many approaches across organisms and even different ML methodologies, including some from our group as well, have been demonstrated in this area. It's overall quite exciting. And there's a very simple way to generate sequences from a masked language model. And this hopefully ties back to the fact that I mentioned, the fact I mentioned about using a uniform masking rate. Since the model is more or less comfortable with any masking or dropout rate, you can then use it in a sequential way to generate sequences from scratch. So conditioned on some specific profile, let's say we want to generate something that's a peak in a specific cell type, we can start off with a completely empty sequence. So you mask all the bases in your sequence, and then you make a prediction with your model. You pick a position, look at the predicted distribution, sample from it, or do something more interesting potentially. Now you selected one base, and then you send that back into the model, make predictions again, choose a position, sample the base, and then repeat this till you essentially generated the full sequence. And you could choose which position to unmask randomly, or you could use the model to guide your position. Where you unmask, you could look at the confidence, for example, from the models, or the 2- entropy to select the next position that you choose to unmask. So to demonstrate this more concretely, we turn to a more interesting case where we have not just one track, but we have 3 tracks coming from 3 distinct cell types. So GM12878 as before, but also dermal fibroblasts and H1 embryonic stem cells, kind of stem cell population. Use train on 1,000 base regions, and again with a uniform masking rate during training. And so once you've trained this model, we can then almost prompt it with profiles that of peaks across cell types that we would like the generated sequence to have in different cellular contexts, right? So here we can prompt it with a cell type specific regulatory element. So we can say, I want high activity in GM1287 that even looks a specific way. So it's somewhat narrow and at the center of this regulatory element, and maybe no activity in fibroblasts and H1ESCs. And then what Nona will do is generate not just one, but you can keep running this multiple times, generate a bunch of sequence candidates that hopefully have the desired functional properties. And then you can pass each of these sequences through a sequence to function oracle model. In this case, just to keep things independent, we use a model like Borzoi. And Borzoi also predicts the profile of activity across the same cell types. So in this case, you can see that upon specifying a sequence to have activity specifically in GM12878s, Nona designs a sequence which is also predicted to have activity in GM12878. So it's kind of fulfilling the design objective, if you may. And we see more or less this across the sequences generated, we see that it's able to recover this kind of cell type specific activity. There are some hits, but there are some misses, but on average, you can see that the generated sequences have GM12878 specific activity in this case. And you can repeat the same thing with fibroblasts or H1ESCs, and it's able to generate regulatory elements specific to these cell types. Or you could prompt it with something that's a bit more ubiquitous. Let's say you want a regulatory element that's shifted to the right, accessible in all cell types, and by and large, it's able to do the same. It's able to design what you ask for. You can also, what we started trying to do is prompt with more complex profile specifications. In this case, you have kind of a bimodal distribution in fibroblasts and a left shifted one in GM12878, but the centers of the peaks aren't really aligned between GM12878 and fibroblasts, and no activity in ESCs. And you can see at least some examples, though not all, seem to recover the kind of expected profile specifications. So this is a prototype of sorts, but you can imagine if we start throwing in other tracks like gene expression or CAGE, you can think of designing entire regulatory domains where you have some desired chromatin accessibility across cell types and potentially even gene expression in different cell types. And we also looked at motifs that are typically enriched within these sequences. So for example, the fibroblast specific sequences that Nona generates, you would find more AP1 motifs in them, which makes sense. AP1 is like the June-Foss family of transcription factors are known to have high activity in fibroblasts. You see similar patterns across the board. The OX-OX motif plays a crucial role in stem cells, regulation in stem cells, and that seems to be enriched in H1ESC specific generated sequences. So to wrap up this section, what we're seeing with functional language modeling is that we're able to learn meaningful regulatory DNA features and also generate sequences with desired profile specs. All right, so I'll start with the last part, which actually touches upon a completely different aspect of genome privacy, which I was shocked was something that we could even get into with these kinds of models. But what I'm going to show you is that we can actually use an instantiation of these functional language models to de-identify attack-seq files, fragment files specifically. Let me show what I mean by that. So if you worked with attack-seq data of any kind, or really any sequencing data, once you get your FASTQ files from the sequencer, you align them to your reference genome, and you end up with a BAM file, which contains information about where each read aligns to, as well as information about any potential mismatches to the reference genome. So in this case, you're seeing that there's an attack-seq peak here, and all the reads from this specific individual contain a mismatch at this position, which suggests that the genotype of the individual here is not the same as the reference here. This individual has a variant at this site, in this case, a homozygous alternate variant. Another way to represent attack-seq, which has gained popularity with the advent of single-cell attack-seq especially, is what we call a fragment file. These are just 3-column, tab-separated files, where instead of supplying a lot of information about the reads, you just specify the chromosome and the start and the end coordinates. So for example, for this specific read, you just say it starts here and it ends here. And there's no information beyond that except just a list of alignment start and end coordinates for each read, right? So in theory, it's de-identified because any information that BAM files contained about mismatches, indels, or other complex sequence mutations is not really captured or is not kept in these fragments in principle. And so the question we asked is if we can actually predict the genotype simply from a list of coordinates, right? So at first it sounds like I'm not even making sense because this information is not there. But if you start digging into ATAC-seq data more closely, you'll see there's actually something interesting going on. So when you look at ATAC-seq, as you might have over longer regions of DNA, you'll see regions with enhanced signal. These are regulatory elements that we've been talking about all morning. But once you zoom into a specific regulatory element and look at, say, 100 bases around it, and when we just look at the start, so if we just look at the starts and ends of these coordinates and count how many starts or ends of any of these files, of any of these reads we have at specific positions, what we'll see is that ATAC-seq at base resolution is actually quite spiky. So what it means is that within this regulatory element of 100 bases, you're seeing more cuts at this specific position compared to the positions around it. So it's not a uniform way of cutting, but it's almost like it's preferring certain bases to cut over others. And the reason behind this is that the TN5 transposase enzyme that's used in ATAC-seq has a sequence preference. What that means is it's more likely to cut at positions where there's a G 4 bases upstream and a C 4 bases downstream. It's not a very hard preference, but it does end up reflecting this kind of spikiness, and it's manifested this kind of spikiness in ATAC-seq profiles. And the recent preprint from my PhD advisor Anshul Kundaji's lab on chrome-BPNet is something you should check out if you want a more detailed treatment of what this bias looks like. But the high-level takeaway is that the TN5 enzyme has a sequence preference, and if you alter the underlying DNA sequence with a mutation, it might alter the pattern of cuts, right? So just to illustrate this in more detail, what we did is we looked at ATAC-seq from the AFGR project, the African Genomes Resource Project, a subset of the 1,000 genomes people. And at this specific variant on chromosome 1, if you just add up the reads from 32 people that have a TT genotype at this specific position, this is the pattern of reads you get. And then there are 15 people that have an AA genotype. And here is the pattern of reads from those people. And both of these have been appropriately normalized. And you can compare the numbers on the left and right panels. And so I'm going to pause for 10 seconds and make this a game of spot the difference. And I hope you can find some positions where there's differences between these plots. It's kind of subtle, but take a squint, I guess. So hopefully you saw a few of these positions where there's differences caused just by the variant, right? So the one my eyes went to immediately was this tall bar here is reduced to some extent here. There's also changes at these three positions. The shape changes from high, low, low to high, low, high. And even around the variant, you see some local changes in the cutting patterns, right? So these aren't actually necessarily variants that affect chromatin accessibility entirely. These are just affecting the local cutting patterns around the variant. So they don't necessarily have to sit within a transcription factor binding site to affect chromatin accessibility. They just can be really any variants within a highly accessible region, right? And so I think it stands to reason that if I were to give you the profiles of around the variant of interest, if you squint at many of these, hopefully eventually you'll be able to predict the genotype from just the patterns, just these sort of cutting patterns of ATAC-seq and nothing else, right? And so to test that out, we trained a Nona model, a very small 128 base long Nona model with the same functional language model loss. So we have the functional tracks here, which is ATAC-seq from GM12878 at base resolution. And then we're masking with a variable masking rate when you're training. And another important point to note is that we're actually just training still with the reference genome. So when we're training this model, we don't require access to any individual specific variants. Just the fact that most positions in the genome don't differ across individuals is enough for the model to latch on to relevant features. And once we're done training, we can go from SNP to SNP, especially in regions with high chromatin accessibility, and try to predict the genotype from the surrounding DNA sequence and the ATAC-seq base resolution signal, right? So once you train this model, I'm going to see if it can actually do exactly this, right? Can it predict the individual variants? And the most interesting positions for one specific individual would be where the individual has a variant that differs from the reference, right? So in this case, we're going after homozygous alternate variants where the reference, for example, says there should be a G here, but the specific individual has 2 copies of NA, for example. And these could also be in the training set, and the model is almost trained with the wrong label with the G here, or they could be in the test set where it's not seen them before. And so we asked, in how many of such cases can the model correctly genotype this individual just from the ATAC-seq cutting patterns? And it turns out, at least in regions with high chromatin accessibility, in the top 1,000 strongest peaks, it's almost as high as 90% for homozygous alternate variants. So we can't really distinguish between heterozygous versus homozygous at this point, but you subset the analysis to homozygous alternate variants. You can see you can actually very accurately predict the individual's variants just from the cutting patterns of ATAC-seq, which were derived from a fragment file, which in theory didn't contain any personal identifying variant level information. So go a step ahead, and let's say we had access to a database of individuals and their genotypes. Let's say there's a malicious actor that's it's collected this, and then we get the ATAC-seq fragment file from Bob. We can go from variant to variant and make predictions at each position. And let's say at this specific position, we have a prediction of 0.7 for C. And we see indeed Bob has 2 copies of C here. Would it be possible to link this ATAC-seq sample from Bob to Bob's genotype? That's the question we asked. And so we can come up with simple scores that match the similarity between each of these genotypes and the predicted probabilities or the predicted genotypes from ATAC-seq. You could simply look at the likelihood of observing this genotype under this model separately for each of these people and then rank individuals that have the highest math score, right? So we did this. What we did then was we looked at 3,200 genomes from the 1,000 Genomes Project. And then for this one specific individual named NA12878, from their ATAC-seq track, we predicted their genotype, and then we tried to match it against these 3,200 individuals. There's a histogram of scores you get. These are math scores of ATAC-seq from NA12878 with 3,200 genotypes. And it turns out the math score of the ATAC-seq from NA12878, the genotype of NA12878 is really high, and it's like a strong outlier. And these are the other individuals in the data set. And what this is really suggesting, it's super easy to identify that the sample came from NA12878 and not the other 3,200 individuals. And so we repeated this analysis for ATAC-seq samples from 83 individuals in the AFGR project and tried to link them against the 3,200 or so genomes in the 1,000 Genomes database. And turns out we can link each sample perfectly, right? So we call this a success if you're able to rank the person from whom the ATAC-seq sample came as the top match for our ATAC-seq samples. And so we're able to do this with almost perfect accuracy for all of these 83 individuals. But if you start subsampling the ATAC-seq within each of these, from the samples of each of these individuals, so you're basically reducing your signal, there is a point of about 5 million reads below which we start losing our accuracy. Or maybe another way to look at it is even if you have 500 cells from some individual and you have a fragment file corresponding to that, it's enough for us to be able to genotype them in this specific situation. Yeah, so to conclude this part, show a Nona model that's able to sort of de-identify ATAC-seq fragment files. And today, if you go on Geo and you look up dataset releases from different papers, you will find papers where FASTQs and other sensitive files have been sent to dbGaP for access control, but you can find fragment files from ATAC-seq publicly available. So that does seem to highlight a potential privacy risk. So these are just 3 applications I covered, but as I mentioned, this is a very flexible framework. You can come up with a mask of your choice and attach that to the task of your choice and hopefully do something really interesting. And we're potentially just scratching the surface of what's possible with this. So Nona is a mass modeling based framework for functional genomics and unifies common use cases like sequence to function and genomic language modeling, but also seems to be able to open up entirely novel applications. And what's really nice is we can flexibly and dynamically change inputs and outputs across modalities, which in this case are DNA sequence and functional tracks. And this is a question that was asked, and in theory, we can support multiple masking modes in the same model, and this does seem to work to some extent, but it's still a pretty interesting direction.